Thank you for visiting the Laboratory of Biology study program. The Biology Laboratory serves as a support facility for students and lecturers' learning and research activities. The laboratory already hosts all of the necessary equipment for the courses in the study program. Students and lecturers can use all of the laboratory equipment. Activities in the laboratory, such as experiment and research completion, have been closely monitored and implementation is aided by laboratory assistants and lecturers. The laboratory has been equipped with facilities or tools to ensure the safety of the laboratory work. Before entering the laboratory community is instructed to put on rubber sandals and then put on laboratory coat to protect our bodies and arms from contact with harmful substance in the laboratory. There are also fire extinguishers in the laboratory, supported by availability of evacuation routes as the most convenient and safety way out in the event of an emergency such as a fire. The laboratory also has a first aid kit as it is a first emergency aid facility. In this laboratory, there is also a safety shower with an eye wash. Other items included in the laboratory's equipment are masks, eyeglasses, helmets, latex and heat-resistant gloves. Then, we'll know about some of the biology laboratory equipment that students commonly utilize for activities and research, which are as follows.
PCR. Students reproduce DNA segment through polymerase chain reaction or PCR using DNA sample. For tools and materials are PCR kit, primer, distilled water, 10 to 100 microliters micropipette, and a tip as a tool for taking small amounts of liquid, and 0.5 milliliters microtubes as a sample containers. Before homogenizing the solution, 3 microliters of aqueous, 2 microliters of DNA sample, 1205 microliters of PCR kit, and 2 microliters of primer were inserted into the microtube and tightly closed. The microtubes are then placed in the PCR, which has been pre-programmed according to the test procedure. Take polymerase, primer, DNA templates, and nucleotides are required to carry out the PCR reaction, or building blocks of DNA. The entire material is placed in a tube, along with the cofactors required by the enzyme. The subjected to repeated heating and cooling cycles, allowing for DNA amplification. Spectrophotometer. The ordinary spectrophotometer, which is used for experiments, and the UV-Vis spectrophotometer, which is used for research, are two types of spectrophotometers. Hitachi U200 spectrophotometer. Students perform a microbiology experiment in which they calculate the optical density value equivalent to macrolide 0.5 on bacterial isolates in psychological sodium chloride media. In addition to spectrophotometer, students use a cuvette, micropipette, spray bottle, hose, Bunsen, and Voltex in this activity. The spectrophotometer examines the level of turbidity or optical density as measured by absorbance value. The wavelength used in this study was 600 nanometer. Based on the preliminary test result, it was determined that this wavelength is the best for reading the density of bacterial isolates in psychological sodium chloride media. UV-Vis spectrophotometer. Students will use a UV-Vis spectrophotometer to calculate the optical density value in WWTP water sample in ammonium test reagents. Several tools, including cuvettes, micropipettes, and spray bottle, were also used. Spectrophotometer for UV visible light. It is a tool that uses spectrophotometer technique to detect ultraviolet and visible light. This tool is used to determine the absorption of ultraviolet or visible light by a solution-based material. The amount of light absorbed by the substance in the solution determines the concentration of analyzed solutions. In this case, Lumber-Beer law can be used to describe the relationship between light absorption and substance concentration in solution.
Electrophoresis. Electrophoresis is used to determine the size and shape of a particle, both DNA, RNA, and protein. This tool is used in the course molecular genetics. The electrophoresis equipment consists of a gel mold for making a gross gel, a well printer for making wells on the top side of the agarose gel for inserting DNA into the gel, an electrophoresis tank, and a current source. ELISA reader. This tool is used in immunology, biochemistry, and biotechnology courses to determine antibody levels in serum. Plugging buffer, sample, ELISA washer, secondary antibody or serum, and substrate colored are the tools and materials used. The aim of this experiment, which was done with an ELISA wearer, was to determine the level of antibodies in serum samples. The first step in measuring antibody levels in serum sample is sample preparation, which includes a control. An ELISA plate coated with antigen stools are multipipette, and reagents is kit. Furthermore, 300 microliters of blocking buffer was added to each well of the ELISA plate that had been coated with antigen before incubating for a hour at room temperature. After incubation, the ELISA plate wells were washed 3 times with 300 microliters phosphate butter saline or VBS, and the wells were tapped during the third washing. Following the completion of the washing process, 100 microliters of the sample was placed in the ELISA plate well and incubated for a hour at room temperature. Following the incubation period, the ELISA plate wells were washed 3 times with 300 microliters PBS. Then, 100 microliters of secondary antibody was added to the ELISA plate well, which was then incubated for a hour at room temperatures before being washed 3 times with 300 microliters of PBS. At the end of this activity, 100 microliters of each ELISA plate was added to each well, then incubated for a hour at room temperature before adding 100 microliters of step solution for 5 to 20 minutes to stop the color formation process. 450 nanometer and 590 nanometer wavelengths were measured with an ELISA reader. The data from the experiment can be copied and viewed on a PC or laptop device using the following table data display. Biological Safety Cabinet or BSC. Students use this tool in antibacterial testing activities, aseptically using any media, E. coli bacteria culture, and antibacterial samples. BSC was used as working room. A petri dish, sterile swab, Bunsen, and tweezers were used. A biological safety cabinet is a laboratory work area with air ventilation designed to protect workers working with material samples, environment, and material samples from contamination or the spread of pathogenic bacteria or viruses. By expelling the air through a HEPA or high-efficiency particulate air filter, a BSC or biological safety cabinet creates an air inflow to protect operators who are handling risky biological samples.
Oven and incubator. This tool is often used for testing component balance, hardening of materials, drying of glassware, and sterilizing. In certain experiments, students would also utilize the oven for incubation. Microbiology, biotechnology, and tissue culture courses, for example. In this laboratory, many types of incubators are available. The oven's operation is nearly identical to that of the incubator. Begin by connecting the oven to the laboratory's power supply. Once everything is in place, hit the on or off button. Wait a few moments for the device display to light up. There's no time to change the temperature by clicking on the temperature button. You can also change the timer to suit your needs. You can now add the sample to be incubated. Wait until the process is finished according to the timing. Autoclaves. Autoclaves are used by students to sterilize laboratory equipment and materials. This tool is used in courses on genetics, microbiology, mycology, and biotechnology. Underland Mayer, bacterial isolates from any medium, and aluminum foil were used as equipment and materials. The autoclave checks the water volume by ensuring that the water level is within a predetermined limit. The material to be sterilized is then inserted. The autoclave is then tightly closed to prevent steam from escaping, as well as the autoclave safety valve. Start the autoclave and set the timer for 50 minutes at 125 degrees Celsius.
Digital vernier caliper. As a result of the antibacterial test, this tool is used to measure the diameter of the inhibition zone of the antibacterial activity in the petri dish. The inhibition zone is measured by measuring the outer zone of the paper disc to the outer limit of the inhibition zone with a caliper. The diameter of the inhibition zone was measured using a digital caliper with millimeters unit. The diameter of the inhibition zone can be measured by a caliper by adding the long and short diameters and dividing by 2. The measurements were taken 3 times by different people who had previously done the perception equalization, and the average was taken. The larger the inhibition zone or bright zone, the more capable the sample is of inhibiting bacterial growth.
Microtome. This tool is used to slice specimen into extremely thin sections for examination under a microscope. In this experiment, little brochure and sample specimen were employed. This instrument is frequently used in microtechnical courses. How does a microtome work? Mounting is the process of utilizing a microtome to cut blocks of preparations. A set of preparations must be made prior to carrying out the cutting. Microtome knife preparation. The microtome knife must be sharp before use that the tissue can be cut correctly and not ripped, resulting in good tissue. The microtome blade is then positioned at the specific angle on the microtome. Position the paraffin block sheet and block preparation on the microtome.
Microscope optilette. Through computer devices, an optilette microscope is used to visualize very small object or micro on preserved preparation. Optilette is a microscope camera that converts an analog microscope into a digital microscope to serve as a tool for documenting microscopic objects. The ocular lens is replaced by an optical camera that is connected to the computer via USB. Optilette can be used to display the object being observed, check the objective lens on the monitor screen, take pictures of what is being observed, record live image, measure the dimension of the object to be observed, provide identification labels or markers, and provide convenience in counting with a counter. Microscopes. Students use a microscope to examine very small objects. This tool is used in almost all experiments involving plant anatomy, animal anatomy, animal structure, invertebrate zoology, and so on. Microscopes come in a variety of shapes and size. Binocular, monocular, and stereo microscope are available. An eyepiece and an objective lens are the 2 lenses in binocular microscopes. Monocular microscopes, which only have an eyepiece, are used to examine details in a cell, while the stereo microscopes is used for low magnification observation of relatively large sample in 3 dimensions. pH meter. Students use this tool to determine the pH of the media. In a 200 ml beaker glass, buffer 7.0, sample solution, and distilled water were combined. Before inserting the electrode to the sample to begin reading, the pH meter is turned on. The pH meter will measure the pH level of the sample after the electrode is inserted.
Centrifuge. This tool is used by students in
microbiology experiment to separate debris in a sample. In addition to using a centrifuge, the practitioner also used a microchip in this activity. Centrifuge separates the particles contained in a solution according to size, shape, molecular density, viscosity of the medium, and the speed of the rotor. Centrifuge utilize centrifugal force, which is rotating force away from the center of the sieve, so that in the liquid, larger particles will automatically move away, while smaller particles will gather in the form of the precipitate in the center of the liquid. The greater the density difference between solid particles, the faster they will move. Meanwhile, particles with a higher density will settle to the bottom and form sedimentation, while particles with a lower density will float on the top. The colony counter. Colony counters are used by students to count the number of microbial colonies on microbial isolates in petri dishes. This colony counter works by enlarging the colony with a loop or by marking some of the colonies in a petri dish with a bullet found on the colony counter. Another word to begin using the colony counter is turn on the general switch. The switch beeps after you adjust the sensitivity knob. Select aim mode, then use the up and down arrows to adjust the lamp brightness or paste color. If you want to increase the brightness when displaying the letter L or light and the brightness value in front of it, press the up arrow or vice versa. Place the petri dish with a square line on the surface. Then, count the bacterial colonies by pressing and marking the growing bacteria with a marker. Resume counting. Press the R or reset button until the display shows the number 000 LI per count button. Fermenter. Students will ferment microbial isolates on a large scale in this activity. A fermenter, a Landmeyer, bacterial isolates from natrium broth media, and aluminum foil were used as tools and materials. The fermenters are outfitted with mechanical and electrical equipment, and some even have a control system for controlling the factor or variables that affect the ultimate goal of fermentation in terms of microbial growth. Potential of hydrogen, temperature, dissolved oxygen, media turbidity, foam formation, and so on are the variables in questions. Terms of use. The fermenter is the tank can be operated aseptically, agitated, and aeration. Then, the operating energy should be as low as possible. Temperature and potential hydrogen must be controlled. Care was taken of the sampling site and excessive evaporation was avoided. Tanks are designed to minimize ferment labor, cleaning, and maintenance.
Micropipette. Students utilize this equipment to correctly transfer small amounts of liquids, reagents, and samples. One specializes in biochemistry and genetics. Tips, reagents, and samples are the items and tools utilized in this experiment. While the use of pipette can be accomplished in the past, among them are prepare the necessary tools and materials, holding the micropipette in a punching motion, place your thumb on the volume control area. Connect the micropipette to the appropriate tip. The solution can be taken by pressing the plunger button and holding it so the solution doesn't spill into any region. By carefully removing the plunger button pressure, transfer the taken solution to a new container. When the process of taking the solution is finished, release the tip by pushing the tip ejector, and tip will be automatically released. Finally, if the tip comes into contact with anything other than the solution in question, it can be changed with a fresh tip. Ceodimeters. This tool is used by students to determine the chemical optical density.
as in the application in microbiology course. The COD meters analyzes procedures to add the sample to the prepared reagents. Then, heat the reagents or sample for 2 hours in COD reactor. Distillator. This device is a distillation machine that extracts a liquid or solid component from 2 or more mixtures of substance based on their high and low vapor points. On a laboratory size, the way the distillation device works with water is to install the equipment accurately and carefully, especially at the connection point, so that there is no leakage. Fume hood. The fume hood is intended to shield people from the dangers of poisonous gas inhalation during the testing process, which is utilized in both research and experimentation. To conduct the experiment, the user must wear safety eyewear and latex gloves. Nitric acid, sulfuric acid, hydrochloric acid, test tubes, and tube clamps were utilized in the fume hood experiment. Front door of the fume hood is used to draw air into the fume hood. The suction blower is then used to remove it. The stuff is then removed from the warehouse or filtered. As a result, the air in the fume hood procedure may mix with the air in the laboratory area. The fume hoods design and operation are centered on preventing the risk of mixing air in the fume hoods with that in the laboratorium itself. You have finally visited the Laboratory of Biology study program. Thank you for your interest in our laboratory and your enthusiasm.